So today it will be on absorption of lipids. So lipids are absorbed. So the glycerol, short and medium chain fatty acids, which are less than 14 carbon atoms, they are directly absorbed from the intestinal lumen into the portal vein and taken back to liver for further utilization. Whereas the long chain fatty acids, the free cholesterol and the beta acyl glycerol together with light salts, they form the mixed micellase. Micellase are nothing but they are disc-shaped clusters of amphipathic lipids which coalesce with their hydrophobic groups on the inside and their hydrophilic groups outside of the clusters. The mixed micellase are soluble in the aqueous environment of the intestinal lumen. The micellase approach the brush border membrane of the enterocytes. So here in this picture, which is in blue color, the round ones, these are hydrophilic polar head group, whereas the long one, that is all this, okay, non-polar hydrocarbon chain. The micellase, you can see in this picture, it is an intestinal cell or the enterocyte. All these are the villi which are thrown into manifolds. These are the villi. And here you can see the emulsion droplet, that is nothing but micellase. All these small ones, these are micellase. So the micellase constantly break down and reform. It is the monoglycerides and fatty acids that are free in solution that are absorbed, not the micellase. Because of their non-polar nature, monoglycerides and fatty acids, they can just diffuse across the plasma membrane of the intestinal cell, the enterocyte. Some absorption may be facilitated by a specific transport proteins. The clinical significance of cholesterol absorption. The drug ezetimibe, which blocks the protein that specifically mediates cholesterol, transports across the apical plasma membrane of enterocytes. Ezetimibe has been shown to be effective at reducing levels of LDL cholesterol, particularly when combined with a statin, a drug that inhibits cholesterol synthesis in the liver. However, several clinical trials have been shown that further cholesterol lowering with ezetimibe does not improve measures of atherosclerotic plaque. Clinical trials, they are in progress to determine whether ezetimibe alone or combined with a statin is beneficial for preventing cardiovascular events. Resynthesis of triacyl glycerol and cholesterol esters. Within the intestinal epithelium, one monoacyl glycerols are hydrolyzed fatty acids and glycerol, and two monoglycerols are reacylated to triacyl glycerols via the monoacyl glycerol pathway. Lysophospholipid are recycled to form phospholipids. Cholesterol is reacylated to form cholesterol esters. Long-chain fatty acids are used for esterification to form triglycerides, phospholipids, and cholesterol esters. Short and medium-chain fatty acids, they are released into the portal circulation and are carried by serum albumin to the liver. Formation and transportation of chylomicrons. So you can all have a glance at this picture. So it is nothing but it is the lumen of the intestine, small intestine. Okay. So here what happens? They are the triglyceride molecules, which are pink in color. Okay, these are triglycerides, along with bile salts, the lipase enzyme. Okay, after action of this lipase, what happens is you get fatty acids, and this fatty acid and mixed molecules with monoglycerides. Okay, this is a combination. So this will enter into the intestinal epithelium cell, that is from the lumen. The fatty acids and monoglycerides are converted into triglycerides, and along with the protein, it forms chylomicrons. And these chylomicrons, they, via the lacteal or the thoracic duct, the chylomicrons enters into the thoracic duct. One of the condition lipid malabsorption is steatorrhea. Lipid malabsorption results in increased lipids, including fat-soluble vitamins A, D, E, and K in the feces. This cause may be pancreatic due to pancreatic insufficiency, including the cystic fibrosis, chronic diseases of pancreas or surgical removal of the pancreas. Then short-end bowel, celiac diseases, screw or Crohn's disease. Maybe bile duct obstruction due to gallstones, tumor of head of the pancreas, enlarged lymph nodes, etc. Milk and coconut oil are used therapeutically since they contain medium chain fatty acids here in treating some of the malabsorption of lipids. Secretion of lipids from the intestinal cell or the enterocytes. Once inside the enterocyte, monoglycerides and fatty acids are re-synthesized into triacylglycerol. The triacylglycerol is packaged along with cholesterol and fat-soluble vitamins into chylomicrons. The chylomicrons are lipoproteins, the special particles that are designed for the transport of lipids in the circulation. Chylomicrons are released by exocytosis at the basolateral surface of the enterocytes. Because they are particles, they are too large to enter typical capillaries. Instead, they enter lacteals, lymphatic capillaries that poke up into the center of each villus. Chylomicrons then flow into the circulation via the lymphatic vessels. Structure of the chylomicron. So this is the structure of chylomicron, which is about the size is 0.1 to 1 micrometer. Average composition of this chylomicron is mostly it is 84% consists of triglycerides. Cholesterol is 2%. Then ester cholesterol is 4%. Then phospholipids 8%, apolipoproteins 2%. Transport and utilization of chylomicrons. So now what happens from the intestinal tract, the chylomicrons, they go via thoracic duct, that is and enters into the venous circulation and systemic arteries, then peripheral capillaries. Then in the peripheral capillaries, especially lipoprotein lipase is acted upon in the endothelium, and it is converted into fatty acids and monoglycerides. Now, this fatty acids and monoglycerides, now it will have 4 fates, that is 1 to liver cells, it is stored in the liver or it reaches the liver, and the other one is skeletal muscle, cardiac muscle, then adipose tissue. This is the way it is being utilized. The chylomicrons are utilized. The clinical significance of chylomicron synthesis and utilization. Defective synthesis due to deficiency of ApoB48 protein. The triglyceride may accumulate in the intestinal cells. Chyluria, which is due to an abnormal connection between urinary tract and lymphatic drainage system of intestines, forming chylous fistula, characterized by passage of milky urine. So that is the significance here. The chyluria, because of chyluria, you come in diagnosing this chylous fistula. Then chylothorax. There is an abnormal connection between the pleural space and the lymphatic drainage of small intestine, resulting in the accumulation of lymph in pleural cavity, giving milky pleural effusion. So chylothorax means you can diagnose it as milky pleural effusion. It gives an idea for you. Then physiologically important lipases. Physiologically important lipases are the lingual or the acid stable lipase, mainly from mouth and the stomach. Here, the triglycerides with medium chain fatty acids, free fatty acid, and diacylglycerols. Pancreatic lipase and colipase, it is from the small intestine, triglycerides with long chain fatty acids. Then, it also acts on giving rise to free fatty acid and diacyl monoacyl glycerol. Intestinal lipase with bile acids. This is from small intestine. It acts on PAG and medium chain fatty acids, giving out 2 fatty acids and glycerols. Then phospholipase A2 and bile acids from small intestine, phospholipids with unsaturated fatty acid at position 2, giving unsaturated fatty acid and lysolecithin. Then next one is lipoprotein lipase. So this is present in the capillary walls, which acts on triglycerides and chylomicron and VLDL, giving out free fatty acids and glycerol. Then hormone-sensitive lipase from the adipose tissue or the cell. It helps in PAG stored in the adipose cells, giving out free fatty acid and glycerol. Summary of lipid digestion and absorption. So in pancreas, lipolysis, the triglycerides are broken down to give you fatty acids and beta monoglyceride. In the liver, the micellar solubilization with bile acid takes place, and the fatty acid and the beta monoglyceride are esterified to give you triglycerides to cholesterol, phospholipid, and beta lipoprotein, that is chylomicron formation. So chylomicron is formed here. This happens in the intestine, especially in the jejunal mucosa, that is absorption after absorption. So next one is then the chylomicron will be utilized to various tissues for various tissues. Through lymphatics, it will be delivered to various tissues for utilization of. So the chylomicrons deliver absorbed triacyl glycerol, that is triglycerides, to the body cells. PAG in the chylomicrons and other lipoproteins are hydrolyzed by the enzyme lipoprotein lipase, an enzyme that is found in capillary endothelial cells. Then monosaccharides and fatty acids, they are released from digestion of triacyl glycerol, and then it diffuses into the cells. Absorption of lipids through the intestinal wall. So you can appreciate this. This is the stomach, the lumen of the, which is continuous, the lumen of the small intestine. A cross section of the small intestine, that is villi of the intestinal mucosa, extended into the lumen. So this is the picture. So here what happens is mixed micellase, they deliver fatty acid to epithelial cells covering intestinal villi. Then intestinal mucosa, they form triacyl glycerols and aggregate into small lipoproteins to form chylomicrons. So this is again a small cut section, small picture showing out clearly all this. Okay, so here what happens from the intestinal mucosa, that is absorption of small chain fatty acids less than 10 carbon atoms directly into the villi of the intestinal mucosa. So here it is being absorbed, and this short chain fatty acids along with that, okay, with the 10 C carbon atoms. Then the intestine and the longer chain fatty acids, they combine with the bile salts to form mixed micellase in the intestine. Then here what happens is this long chain fatty acids and bile salts, which gives rise to formation of mixed micellase, and this mixed micellase will enter into the villi. So each of the villi of the small intestine, they consist of a layer of epithelial cells. They contain epithelial cells and a core of capillaries, and connective tissue. The outside apical face of each epithelial cell is covered by a smaller projections called the microvilli or the brush border. The fatty acids, they diffuse across the brush border membrane and into the epithelial cells. There the fatty acids combine with glycerol to form a new triacyl glycerols, which aggregates with lipoproteins forming chylomicrons. Taken up by the capillaries, chylomicrons circulate to the liver and other organs. Thank you.